stiffness in your environment rewrites who you fundamentally are. It's not just about psychology, it is physics at the cellular level. Every tiny piece of tissue, the microscopic scaffolding called the extracellular matrix, is constantly transmitting tension signals to our cells. These signal hubs use molecular anchors we call integrins to translate mechanical stress into chemical instructions inside the cell nucleus itself. Imagine your body as a perfectly tuned instrument. If the surrounding material is warped, the pitch changes completely. That subtle physical shift dictates gene expression and ultimately how stable or coherent you feel internally. We are building a framework that finally connects this deep biophysical grounding to subjective states of self-presence. Imagine your body not just as a collection of cells, but as an impossibly complex architectural blueprint built from protein strands. The environment around these cells, the extracellular matrix, isn't passive scaffolding. It's constantly sending physical instructions. When forces shift on that matrix, things like the subtle tension in connective tissue trigger specific molecular events inside the cell. This mechanism is called mechanotransduction, essentially a cellular language translator. It converts pure physics into chemical messages. Think of it as tiny stress sensors determining whether the whole system should stabilize or restructure itself completely. That ability to sense and adapt reflects an unbelievable biological self-attunement, allowing systems resilience at its most fundamental level. It's not just about cells sticking together anymore. Imagine your entire body as a complex machine that can literally sense its environment, the stiffness, the tension, the subtle pull from tissues around you. These connections aren't passive anchors. They are tiny sensors translating external physical force directly into internal cellular commands. We found these microhubs of connection lead to fractal resonance. The way your tissue self-organizes creates a unified field sense within your body. It implies that stability or grounding itself isn't just mental, it's profoundly architectural. Which means something nobody expected for how we define internal coherence. It's not just that cells adhere to their environment, they actively listen to it. Think of your body like a finely tuned violin string. The subtle tension from the matrix isn't passive scaffolding. It's an active signal telling the cell exactly what its job is right now. This dynamic feedback loop means our fundamental resilience comes directly from how well we interpret physical constraints through mechanisms like focal adhesion kinase signaling cascades. We are literally self-attuning ourselves based on structural information. Some researchers argue that these systems collapse under even minor stress, but others show robust homeostatic consciousness at work. How do you manage the internal chatter when your very foundation is vibrating? That leads us to where perception and physics meet. Think about your body not as an organism, but as a vast piece of incredibly sensitive physical hardware. It's constantly feeling tension, pressure, the stiffness of the material around every single cell. Integrins are these microscopic sensors that translate shear force into biological information. When they cluster, they aren't just holding things together, they are generating complex electrical signals across the system. This is mechanotransduction at its highest level. It's like a massive distributed sensor network reading geological stress and turning it into personal chemistry. The receptor fires. It tells the cell what the environment feels like. But what happens next, how that signal organizes itself to influence your internal state? That's where resonance comes in. We always thought biological structure was just scaffolding for function, like building supports holding up an observation deck. But what if the support itself isn't passive? What if it actively dictates who we are? The data suggests that how tightly our cells grip their environment, this process of integrin clustering doesn't just affect growth or division. It influences systemic coherence. Imagine a tuning fork vibrating not only when struck, but in response to ambient pressure changes across the room. That's what is happening inside us. Our structural integrity may actually be generating and modulating our subjective sense of presence, our grounding. But if that connection exists, then we are missing something fundamental about how tension translates into thought itself. Think of your body like a highly sophisticated architectural structure. Its stability depends entirely on how tightly those foundational connections hold together. It is this subtle, continuous negotiation between internal tension and external environment, a process called integrin mechanotransduction, that tells your cells whether to maintain resilience or enter collapse. When cellular anchors cluster correctly in the extracellular matrix, they are essentially tuning themselves into a coherent state of being, like finding perfect resonance with its surroundings. This shows us that groundedness isn't just a mental concept, It is a physical architecture we build and maintain moment by moment through biological feedback loops. Look for systemic coherence, not interventionist fixes, but structural integrity in your own life. The full research archive is at jmthomasofficial.com. The architecture of your body is a continuous conversation between force and meaning. When those tiny adhesion points, the integrins, are stressed just right, they tell the entire system how to be grounded. Think of it less like a circuit board and more like root systems in fertile soil. Structure dictates flow. This constant feedback loop, this biological self-attunement, is what defines presence. Your resilience isn't abstract. It's physically rooted in your matrix integrity. To enhance that coherence, focus on finding consistent biophysical stability in your immediate environment, allowing the system to settle into its most cohesive state. The full research archive is at jmthomasofficial.com.